Good morning, everyone. Good morning, everyone. This is the public meeting for the Amyloid Cardiomyopathy ATTR public meeting. At the beginning of every Midwest CPAC meeting, we want to ensure that all speakers disclose any financial relationships with industry. Every member of the Midwest CPAC panel who's present today has met the ICER conflict of interest policy. I'd like to introduce all of the Midwest CPAC members, or let them introduce themselves. I will call on them. Please mention your work and note whether you have any updates to this declaration regarding your conflicts of interest. I'll start. My name is Jill Johnson. I am a professor of pharmacy practice in Little Rock, Arkansas at the University of Arkansas for Medical Sciences, and I have no further conflict of interest to report. Eric.
My name is Eric Armbrook. I'm a professor at St. Louis University. I have no conflicts of interest to report.
Alan.
Hi, I'm Alan Balch. I'm the CEO of the Patient Advocate Foundation, as well as the National Patient Advocate Foundation. I have no updates to my conflict of interest and apologize to everyone. I'm not able to participate by video this afternoon due to an unforeseen family issue.
Thank you. Bijan.
Yeah, hi everyone. Good morning. I am Bijan Bora. I am a professor of health services research at the Mayo Clinic College of Medicine and Science, and I have no updates to the conflict of interest as defined by ICER. Thank you.
Aaron.
I'm Aaron Carroll. I'm a pediatrician and health services researcher. Currently, I'm the president and CEO of Academy Health. I have no updates to my conflicts of interest.
Gregory.
Good morning. I'm Greg Kerfman. I'm the executive editor of JAMA. I'm a cardiologist by training, and I have no conflicts.
Inge.
Yes, hi, I'm Ingve Falketer. I'm a professor of medicine at Case Western Reserve University in Cleveland. Thank you for having me. I have no conflicts of interest.
Heather.
Good morning. Heather Guidon, Program Director of the Center for Endometriosis Care and a board-certified healthcare advocate. I have no updates and no disclosures.
Gianni.
Gianni Jawadana, a health economist and an associate professor at University of Kentucky. I don't have any further conflicts of interest to report.
David.
Good morning, David Kim, a health economist, assistant professor of medicine and public health sciences at University of Chicago. I have no conflict of interest.
Brad.
Hey, good morning, everyone. I'm Brad Martin, professor in the Division of Pharmaceutical Evaluation and Policy at the University of Arkansas for Medical Sciences, and I have no updates to my conflicts.
Timothy.
I am Timothy McBride. I'm a professor at Brown School at Washington University, and I have no updates to my conflict of interest.
Rachel.
Rachel Sachs. I'm a law professor at Washington University in St. Louis, and I have no conflicts of interest to report.
Kurt.
Good morning. I'm Kurt Vandenbosch. I am a pharmacist with the St. Luke's Health System in Idaho, and I have no conflicts of interest.
Stuart.
Good morning. I'm a clinically retired cardiac electrophysiologist, and I currently work as a patient experience consultant for a large multi-specialty group from Trinity Health in southeastern Michigan, and I have no conflicts of interest. Thank you.
Okay, did I leave any Midwest CPAC members off? Anyone left? Now I'd like to call on individual patient and the clinical experts to introduce themselves and declare any potential conflict of interest. The first one is Milton Mitchell.
Yes, Milton Mitchell, patient advocate, donate life ambassador. I have no interests or problems with money. I'm retired.
Thank you. Sean.
Yes, my name is Sean Riley, a hereditary amyloidosis patient, treated at Brigham Women's Hospital in Boston. I'm a speaker for the Amyloidosis Speakers Bureau, and I have no updates to my conflict of interest statement.
Dr. Michelle Kittleson.
Hi, I'm Michelle Kittleson. I'm a heart failure transplant cardiologist and professor of medicine at Cedars-Sinai in sunny Los Angeles, California. I have no relationships with industry or conflicts of interest to disclose.
Thank you. Dr. Matthew Moore.
Yeah, hi, Dr. Moore, professor of medicine and cardiology at Columbia University. I have no updates to my conflicts to disclose.
Okay, thank you. Thank you all for being here. Now I'd like to introduce Sarah Iman, the president and CEO of ICER.
Jill, thank you so much. My thanks to Jill and the entire Midwest CEPAC for coming together today for this important topic. I am the president and CEO of ICER. I'll be joined today by several of our fellow ICER employees and consultants. You'll hear from Jason Waspy, who was the evidence author for this report. David Rind will be chiming in, our chief medical officer who served as the senior management team lead. Stephen Pearson, special advisor and founder of ICER, will moderate the policy roundtable later today. And Aaron Nguyen was the lead modeler of this report, and we'll hear from him a little later. Next slide. Every time we come together to talk about new interventions that we've looked at from a comparative effectiveness perspective, we want to remind everyone that what we're doing is thinking about the lived experience of the patients with this condition. As part of our review, we spend time talking to patients, patient groups, their caregivers, and this is a quote that really stood out to us when we did those conversations. When complaining for years and years, everyone sent me to a different specialist until my cardiologist saw my AFib going into effect. I got tested by tons of doctors until they finally got me diagnosed. I have neuropathy in my feet. It's very hard for me to walk a couple of 100 feet. Still go out with all my friends. They just know I walk slow. I'm living with this. Everybody I know knows I have it, and they are really accepting. They do everything they can to help me out and take me everywhere I need to go. The impacts of this cardiomyopathy condition will be a theme that we talk about today and how these new therapies might address it. But we think about the way it impacts individual patients, their families, and their communities. And again, that'll be a theme that we revisit through all of the conversation today about the evidence and value. Next page. So what do we do when we have a new therapy? There's a lot of different things that are going through everyone's mind when there might be a new therapy for a condition, especially a serious condition like this. And what do we know when it goes towards FDA approval? Well, of course, there'll be some celebration if these therapies, the two that have not yet received approval, get approved, because having more innovation to treat serious conditions is what we all want out of our biopharmaceutical innovation ecosystem. But we don't have all of the answers that we need on that day. We're going to have remaining questions about the risks and benefits. How does this treatment work in the real world? What do we know about the populations that might benefit most? How do these new treatments fit into what we already do? FDA is asking questions about safety and efficacy. We're here to talk about things like effectiveness. What does it mean to have this new therapy and potentially added on to existing therapy? And what are the reasonable prices and costs that we should think about for these new therapies? Cost to patients, the health system, and public payers. And we also love to spend time thinking about what we don't know. As a bunch of academics and researchers, those questions are also really important. Where are the evidence gaps? Is it about particular patient populations? Is it about long-term efficacy? And we'll touch on those in our policy roundtable today so that we can help guide future research for therapies for this condition. Next slide. But when we think about this question of comparative clinical effectiveness, comparative value, we think also beyond the patients who have cardiomyopathy. We think about all of the patients and all of the people that are impacted because of rising healthcare costs. So we think about the wards of Chicago who were left $80,000 in debt after the birth of their twins because they were premature. We talk about and think about Sherry Foy, who was forced into bankruptcy because she needed emergency surgery to remove her colon. And the Bucks of Arizona, who tried to see a doctor and were turned away because they owed the doctor money, forcing them to seek expensive emergency care. And recently, the state of Delaware told the world that they needed to raise healthcare premiums 27%, largely because of rising costs of outpatient medical procedures, emergency room visits, and specialty drugs. Another theme that you will hear throughout the conversation today, even though we're going to go into the weeds about these new therapies for cardiomyopathy, is we are going to talk about other people in the healthcare system and how we can ensure affordability and access for everyone, not just people with this particular condition. Next page. And that's because we like to think about not only the patient in the doctor's office with a particular condition and the importance of that relationship and that they have access to the treatment they need, but we're making choices about price and access that have the least amount of harm to everyone else in the healthcare system, whether it's because of rising premiums or more cost sharing from benefit design. That's the balance we try to reach when we talk about what a fair price is for new innovations. Next slide. So ICER, Institute for Clinical and Economic Review. We are the ones responsible for the conduct of the evidence review that's under discussion today. Our Midwest CEPAC, who introduced themselves at the top of the hour, are independent experts, and you heard their background: health economics, patient advocacy, evidence-based medicine. They're here to help understand how the evidence of effectiveness and value of these new therapies fit into our broader conversation about price, access, and affordability. And we're thankful for their time today. Next slide. We invited everyone to share their conflicts of interest at the top, and we also disclose ours. All of ICER's research is funded by nonprofit foundations, the largest of which comes from Arnold Ventures, but also the California Healthcare Foundation, the Commonwealth Fund, and the Donahue Medical Research Foundation, and the Peterson Center on Healthcare. We also take money from manufacturers and health plans, but that money is firewalled off to support the policy summit that we hold every year for our policy leadership forum, where we try to tackle some big issue in health policy. Our most recent was how to pay for gene therapies. Put that out in April for anyone who is interested. Next slide. This took 8.5 months to get to this point. We started by scoping the research questions that we wanted to ask through the development of this report, and we did that by talking to patients, their caregivers, clinical experts, manufacturers, and other stakeholders. The evidence synthesis itself was led by Jason Waspy from Harvard Medical School, and Aaron Wynn from the University of Illinois at Chicago did the economic modeling. Once the draft report was up and we received public comment, we also sent it out for expert review. Those expert reviewers are listed here. And the public comment informed a revision of that report that forms the basis of the conversation today, is meant to support this deliberation of the Midwest CEPAC. Next slide. So when you're thinking about, if you're following along at home and you've got the report there are the key parts of the value assessment framework that we try to address throughout the report. The stuff in green is the stuff that's more easily quantifiable. If it's a life-limiting condition, does this therapy help you live longer? Even if it is or is not life-limiting, what is it doing to this, your side effects, to your symptoms? Is it improving your quality of life? And what do we know about how much we're spending and how much we might be saving by a new effective treatment? We'll also have a conversation today about the things that are harder to quantify in the blue boxes: special social ethical priorities, benefits beyond health. There's certain reasons that we might want to invest in a new therapy because it might address a health disparity, or it might be for a special population like pediatrics. Those questions will be part of our conversation today. And on the next slide, you can see what we're going to do today. You've made it through the convening and the opening remarks, and we'll be heading on to the presentation of the clinical evidence with Dr. Waspy in just a minute. Then we'll talk about the economic model that we developed and the results of that model, and we'll have time to hear from the public and what they wanted to share for the Midwest CEPAC as they consider all of the different information in their deliberations. After lunch, we will have that deliberation and vote on the evidence. We'll take another break and then come back for a policy roundtable discussion. That discussion is meant to say, okay, this is what we know about the evidence of both effectiveness and value. What does this mean for our policy making and our policy decisions? We'll end with some reflections from the Midwest CEPAC before we adjourn. One quick note before I hand it off to Dr. Waspy: if you are following along at home and have the evidence report up, The background section will give you a sense of what this condition is that we're talking about today. We'll definitely highlight the patient and caregiver perspective section that came from talking to numerous patients and patient advocacy groups about the lived experience of this condition. Then the comparative clinical effectiveness section talks about what we know about the evidence of these new therapies. And then the long-term cost effectiveness section is the economic modeling section. You also might want to look at the benefits beyond health and special ethical priorities that'll inform some of the voting that will happen later on today. With that, it's my absolute pleasure to pass things off to Dr. Wasfi, who will bring us through the presentation of the clinical evidence.
Thank you so much, Sarah, and thank you so much to all of you for being here. Jason Wasfi, I'm an associate professor at Harvard Medical School, and I direct our program in outcomes research at the Mass General Cardiology Division within Mass General Brigham. Next slide, please. This work is mine, but it's also a team. Dimitri Ninikin and Finn Raymond, Emily Nahn have made enormous contributions to their report, and I'm so grateful for their work on this. Next slide, please. Transthyretin amyloid cardiomyopathy is a heart and muscle disease that's caused by amyloid fibril deposits that go into heart tissue. Essentially, this normal physiological protein that has a normal physiological purpose misfolds and deposits into heart tissue. That can happen with two main mechanisms. One is a hereditary version where there's a mutated gene that causes it more likely to misfold and deposit. And the other is a version that happens with age, which is a little bit more common. The exact prevalence is really unknown. Some estimates suggest 50,000 Americans, 150,000 Americans might have it. There are some reasons to think it actually could even be much higher. ATTR-CM is a progressive disease with high morbidity and poor life expectancy, and often exists in conjunction with other disorders like neuropathy because amyloid can deposit in different parts of the body. Next slide, please. Patients face significant challenges, especially in obtaining accurate and timely diagnosis. Essentially, every patient you talk to, and we talk to many, have a story like this. Clinicians are often unfamiliar with the condition. There's underdiagnosis and delayed diagnosis that can be many years. Employment and activities of daily living are hard. They're hindered by symptoms like shortness of breath, arrhythmias, heart failure symptoms. And there's tremendous problems with high medication costs. They're burdensome. There are rebate programs, but they're very difficult to navigate. And again, every patient has a different story about the difficulties in the pathway with these patient assistance programs. Next slide, please. In referencing this talk, it's important to introduce the NYHA functional classification, which is a general clinician-assessed typology for heart failure. Class 1 symptoms are essentially no symptoms, but they have structural disease. Class 2 symptoms are slight limitation of physical activity. Class 3 is marked limitation. So there's a really big difference between Class 2 and Class 3 in terms of symptomatology. And Class 4 has symptoms even at rest without exertion. And that'll be important in thinking through the talk today. Next slide, please. The standard of care involves the goals of relieving symptoms and slowing disease progression. When I trained as a cardiology fellow, there was really no treatment for this. If people were young, you would often consider them for transplant, but there wasn't much aside from that. The landscape has tremendously changed. Current heart failure guidelines recommend the use of tafamidis, which has become available about 5 years ago for individuals with wild type or hereditary, even in these two main variants of ATTR-CM, and symptoms between Class 1 and Class 3. There are alternatives, especially in the overlap situation where polyneuropathy is also present. Next slide, please. The interventions that we consider today are tafamidis and acoramidis, which are different types of ways of stabilizing transthyretin and therefore reducing its deposition into the heart. We're also going to look at vutrisiran, which works through a different mechanism. It silences the production of the protein, which is a slightly different mechanism than the stabilizers. Next slide, please. The scope of the review includes adult patients with ATTR-CM, either wild type or hereditary, again, the two main versions that form the vast majority of people with this condition. The interventions are tafamidis and acoramidis, the stabilizers, and vutrisiran, the RNA blocker. Each comparator is compared to one another, and also each comparator is compared to no disease-specific treatment. Given evidence that has become newly available in the past month from the HELIOS-B trial, we are also making comparisons of vutrisiran plus tafamidis, which is the only drug that's FDA labeled for this now, versus tafamidis alone. Next slide, please. The key outcomes we're looking at is all-cause mortality. In general, in these trials, transplant or ventricular assist device are treated as deaths. Cardiovascular-related hospitalizations, there's subtle differences in ways that the trials define that. And primary endpoints, which are composite endpoints of multiple outcomes that include all-cause mortality and cardiovascular-related hospitalizations. Next slide, please. In terms of the clinical evidence, most of the clinical evidence comes from three key trials. The ATTR-ACT trial evaluated tafamidis. This was the first trial that was conducted. The ATTRibute-CM trial evaluated acoramidis, and the HELIOS-B trial evaluated vutrisiran. The inclusion criteria, as you can see, there's subtle differences, but they're relatively similar. But as you'll see, the study populations enrolled for complex reasons that we'll go into ended up being very different. Next slide, please. In terms of the baseline characteristics, you can kind of see what I mean. Even though the inclusion criteria were similar, they were conducted at different times. So the ATTR-ACT trial, there was no real therapy for this. The patients were much more advanced. The diagnosis wasn't as early because of advances in imaging methods. And you can see that there are many more class 3 heart failure patients in the oldest trial, ATTR-ACT, evaluating tafamidis. And then it goes down for ATTRibute-CM, which evaluated acoramidis. And then it goes down even more in the evaluation of HELIOS-B. And you can see the years. This correlated with progressive years in terms of the conduct of these trials. Next slide, please. This is another way of looking at this point. If you look at survival of patients in the placebo arms of these trials, you can see that they're very different. Speaking to the different study populations, they survived less in ATTR-ACT in the placebo arm, a little bit more in ATTRibute-CM, and then more in HELIOS-B. The other thing that's going on here, the patient population is shifting, but there's something else, which is that tafamidis was the first drug. So as you do new trials, it's a randomized comparison of the new thing. Tafamidis, people on individuals with tafamidis are in the trials in both arms of the trial. But there seems to be a synergistic effect of both of these things. You can see when we separate the monotherapy people in HELIOS-B versus the overall, that this effect of a healthier population with subsequent trials, or I should say a population that does better, is in part due to patient characteristics and probably earlier diagnosis, and also in part due to the fact that an effective medication, tafamidis, is being given in both arms of the trials. Next slide, please. In terms of the results of these trials, tafamidis shows a better survival than placebo when they group together two groups of individuals getting 20 milligrams and 80 milligrams versus a placebo with a hazard ratio of 0.7 and a statistically significant result. For acoramidis, this is a little bit more difficult to assess for complex statistical reasons. There was a trend towards improved mortality, but it was not statistically significant. Hazard ratio isn't reported because of a violation of the proportional hazards assumption, basically the shape of which the curve diverges. There were sensitivity analyses done in other ways, but suffice it to say, there's less clear evidence that acoramidis is associated with reduced mortality. For vutrisiran, again, these data are recently published and released at the ESC meeting this month, or at the end of last month, that demonstrated that vutrisiran is associated with reduced improved survival, reduced mortality with a hazard ratio of 0.71. And interestingly, there's no effect of treatment effect heterogeneity within the individuals. About 40% of the individuals in both arms were getting tafamidis. So this appears to have an improvement on mortality, whether or not an individual is also receiving tafamidis. Next slide, please. In terms of cardiovascular-related hospitalizations, in tafamidis, there was a reduction with treatment of relative risk of 0.68. With acoramidis, again, a reduction, relative risk of 0.5. And then in vutrisiran, you see a similar effect, again, both in the individuals receiving tafamidis and those individuals not receiving tafamidis, with no evidence of heterogeneous treatment effect. Next slide, please. In terms of the comparing of these medicines, this is really interesting. And the one-liner here is that you can make an argument for any one of these medicines probably being better than the others, and it kind of depends on what you want to focus on. In terms of acoramidis and tafamidis, a lot of the argument is around the degree of stabilization of TTR. Again, these aren't randomized comparisons. They're secondary analyses of randomized data. But it appears that increases in TTR levels, which is to say greater stabilization, is associated with improved outcomes. It's difficult to say. You can see here that that there was at least a conference abstract for the retrospective cohort study that compared people who were getting acoramidis in the trial versus tafamidis in real-world practice. We have limited confidence in the importance of these comparisons. It's very difficult to compare different trial populations to real-world populations. And again, acoramidis hasn't yet been used in real-world practice, and vutrisiran hasn't been used for this indication yet. So it's difficult. It's very difficult to make these comparisons between the drugs. Next slide, please. In terms of vutrisiran versus any of the stabilizers, again, there's no head-to-head comparison. So it's very difficult to say, and it's very difficult to, I would discourage people from sort of extrapolating the hazard ratios from the individual trials and comparing them against one another, because again, that slide was very important about the mortality differences. These are all different populations that are seen in the different trials. There's also an interesting issue about tafamidis 80 milligrams versus tafamidis 20 milligrams in terms of comparative effectiveness. There are some data suggesting that 80 milligrams relative to 20 milligrams might be associated with reduced mortality, but it comes from a long-term extension from the original trial, and there's some complex issues around imbalance, potential imbalance, at least in the patients transitioning between different phases of the trial, as well as the timing where a formulation of tafamidis was changed. So I think it's difficult to put too much confidence in these data talking about 80 milligrams versus 20 milligrams. There's an adjunct argument that went into the FDA decision to approve it at 80 milligrams, which has to do with the amount of stabilization, a mechanistic conceptual argument that many clinical experts think is important. Next slide, please. In terms of harms, these are pretty safe drugs. Across all the trials, the incidence of adverse events was similar in the interventional placebo groups. We don't see a lot of side effects or adverse events with any of these medications. Vutrisiran has been associated with specific adverse events like joint pain, reduced vitamin A levels, which would be expected given the mechanism of the drug. And the FDA-approved label for vutrisiran, again, it's approved for a different indication, recommends supplementation of vitamin A when you're taking it. But overall, we're not too concerned about harms of any of these medications. Next slide, please. In terms of heterogeneous treatment effects and subgroup analyses, there's not much here. For tafamidis, there was a slight increase in CV heart hospitalization, cardiovascular hospitalization for a specific NYHA3 group, which is sort of paradoxical and not really consistent with the other data. It may be due, as the authors argue, which is compelling, is that due to longer survival with tafamidis in that group. Acoramidis did not have any big subgroup effects in any specific area. And vutrisiran, again, there were no large, the effect seems to be pretty consistent throughout all groups. There were some greater improvement in younger patients and those with lower baseline levels of NT-proBNP, but again, all groups benefited. So there wasn't any large indication of heterogeneous treatment effects. Next slide, please. In terms of controversies and uncertainties, tafamidis, there's questions. That was the first trial. It's an older trial population. People were diagnosed in different ways. There wasn't an existing treatment that was effective. So there remains an unanswered question about whether tafamidis is effective in a contemporary population that's probably diagnosed earlier. The argument against that being a concern, just to fully articulate all of this, is that the subgroup analyses didn't show any concern, which is to say that in ATTR ACT, the lower NYHA classes, the patients that were in better condition clinically, seem to have benefited the same way. But it is worth noting that it's perhaps a necessary consequence of tafamidis being the first drug. It hasn't been evaluated in a more recent clinical trial. There are some real world data, but it's from registries, but it's difficult to make comparisons because of things like selection bias. Comparing tafamidis and acoramidis is really difficult. You can see in the public comments that we received is both manufacturers kind of have an argument for their medication being better. A lot of this is about the relevance of degree of stabilization at TTR levels as a surrogate marker for outcomes. We've asked clinical experts about this, and people have different opinions about whether this is going to be seen as something that's linked to patient-important outcomes later. I do think there needs to be further thought about the comparison of the trial populations. Randomized controlled trials are amazing for assessing comparative efficacy, but they only apply to the population that's tested. And again, these trial populations are different. It's a good story. People are getting diagnosed earlier and more often treated with tafamidis, but it creates a situation in which the patients are less sick over time. There needs to be more data about combination therapy. So the results from HELIOS-B, because about 40 of the patients of the individuals, again, in both arms, placebo and vutrisiran, were taking tafamidis, and there wasn't evidence of a heterogeneous treatment effect. It suggests, but not proves, that combination therapy from a clinical efficacy perspective might be a compelling pathway. For acoramidis, because of these sort of complex statistical issues, a mortality benefit of acoramidis was difficult to show. Again, it was a more difficult task because the trial population is getting healthier and healthier with time. But then in HELIOS-B, vutrisiran demonstrated a large, statistically significant reduction in mortality in an even less sick population. So that was sort of impressive, although we acknowledge that the trial was longer. So there's sort of complex issues in terms of the evaluation of these results. My overall opinion, and we'd love to talk about this more, is that it's very difficult to compare the comparative effectiveness of these three agents against one another. There is also some uncertainty around tafamidis dosing. The FDA-approved label is for 80 milligrams, but we have found evidence, and this was with extensive conversations with patients, that in some formularies, they kind of push towards the 20 milligram dose, even though that's not the FDA-approved label. It can be given because the pills are produced in 20 milligram pills. There's some suggestive evidence of greater stabilization with 80 milligrams, and then there's also some outcomes evidence, but again, it wasn't preserving full randomization. So it's an area of controversy that I think would be interesting to explore in today's session. Next slide, please. In terms of other benefits and special ethical priorities, one thing that's so clear from our 6 months on this is that ATTR-CM is widely underdiagnosed. So there needs to be better efforts about risk stratifying individuals for screening and sort of more awareness among providers and potentially even different ways of delivering and diagnosing. Every patient with this has a story about delayed diagnosis. And then also the one available therapy for the moment that's FDA approved is very expensive. So there's a lot, patients come to us with a lot of concerns about the pathway to getting treatment that they need. Black patients in the United States are underrepresented in clinical trials compared to white patients, despite the fact that they're more affected, at least by the hereditary version, not necessarily the wild type version. Another matter to consider is that this is overwhelmingly a disease of men, but and all patients are underdiagnosed, but women are probably more underdiagnosed. It's around complex issues around the way that people are screened for amyloid and the sort of heart wall thickness and whether that's gender normalized or not. So even though it affects more men, women are probably even more underdiagnosed than men. We do think that there's compelling evidence. These drugs all seem to work, that these new therapies could reduce the burden on caregivers, which are very substantial. Acoramidis and tafamidis are oral medications, and vutrisiran is subcutaneous. We have no real reason to think systematically that one or the other will be better for access, but it is worth noting that they're different mechanisms. Next slide, please. In terms of public comments, one, for example, was the comparison of the two stabilizers is difficult, but both manufacturers have a case for why their drug is probably more effective. And there are many data gaps, including the impact of treatments on patients with multi-organ involvement. Again, because of the way that the pathophysiology of amyloid, it deposits in multiple organ systems, and people have overlap syndromes with non-cardiac symptoms and conditions. Next slide, please. Overall, it's an exciting time in amyloid. 3 disease-modifying therapies have shown significant potential in reducing mortality and cardiovascular events in individuals with ATTR, TTR, CM. The comparisons between these drugs is really difficult. Again, there are arguments one way or the other, but to me, that's sort of the definition of equipoise, that it's not clear which one is better than the other. All of these agents have a very favorable safety profile. We're not concerned about safety in any of them. Next slide, please. In this setting, we've given B+ ratings. These are strong ratings to acoramidis and tafamidis versus no disease-specific treatment. And we've upgraded the rating for vutrisiran either as opposed to current therapy alone or no disease-specific treatment, with current therapy alone sort of meaning tafamidis, to an A rating. I caution about any interpretation of the difference between an A and a B+. These are all strong ratings and really has to do with the relative degree of certainty we have about the lower bound of the magnitude of effect. This shouldn't be interpreted as a comparison of the different agents against each other. We think it's insufficient, all the comparisons between the medicines, which one is better than the other. Next slide, please. We'd love to take questions, and I'm sure that clinical experts could weigh into depending on the nature of the questions. Thank you all for listening to my talk, and thank you again for our team. And thank you for all the patients who met with us over these last few months, and the trialists and the experts and the representatives from the pharmaceutical companies. You really have enriched this long report with tremendous meaning, and I really hope that we can make some progress today to help these individuals. There's a lot of new therapies in the arena, and we'd like to see them used well.
Jason, thank you very much. Outstanding work on the review for you and Dimitri. The Midwest CPAC has about 20 minutes for questions. If you would push the raise hand, and I'll call on you if you have questions you want to ask about the clinical evidence. Bijan.
Yeah, thanks so much. I guess my question is about the sort of very wide age range. So I see that it is from 18 to 85 or 90, I think, depending on which drug they considered. So my question was, I think if I understood it correctly, it seems it is typically a disease for older patients, and of course there is other group which is which can be hereditary. Now, in my mind, there will be a lot of younger patients in the second group, meaning because it's hereditary for them. And then there will be a lot of older patients from the other group. So my question is, in terms of really kind of combining these two very distinct patient groups in the trials, how is the evidence sort of mixed up in here? I mean, are we really getting sort of because remember, there will be a significant proportion, again, it's my guess, there will be significant proportion of younger patients because of their hereditary conditions versus there will be a lot of older patients that, that would have this condition, and they, because of their older age, besides this condition, there, it can, the condition itself can be actually, would be influenced by the other, other comorbid conditions that these patients might have, like the second group has. So now my question to the group, the clinicians expert here is, how should we be taking the evidence from these trials, given the fact that these two groups are sort of, part of the trial?
I think I probably should defer that to Dr. Moore and Dr. Kittleson.
Yeah, I think it's a good question. The most common variants in the United States are two. One is called V122I, which disproportionately afflicts individuals who have West African ancestry and self-identify as either Black or Caribbean Hispanic. And the second most common is what's called the Appalachian variant, that's THR60 ALA. Notably, while your supposition is correct that they're usually younger if they have variant disease, this disease has an age-dependent penetrance. So most individuals who are still affected with those two variants are not younger than the age of 50. So they could be disparate. The average age, for example, of a wild type patient at our institution who's diagnosed with non-genetic disease is now about 78 or 80 years of age, but a variant patient is in their lower 70s, maybe late 60s. So they're all still quite old if you put in that perspective. And the data about the therapies, I would just comment, seems to demonstrate efficacy whether you have wild type or variant disease. Variant patients do progress at a faster rate because of their variant nature. It's clearly associated with a unstable TTR.
Thank you. David.
Yeah, thank you for a wonderful review. I have a questions about the harm. So I think during your presentations, you mentioned that all these agents are not much associated with the harms, and then pretty much similar across these agents. But in the table 10 in the evidence report, where it shows the key trial harms, and when I look at the rows with the severe TEAE, which is treatment emergent adverse event, the ATTR Act has a pretty similar proportions of populations reporting the severe TEAE, 62.5% versus 64.4%. When you look at the attribute CM, the acronyms reported 37.3% versus the 45.4% in the placebo arm. So if you're looking at like rate ratio, that's the tafamidis almost close to 0.97 versus the acronyms close to 0.80 and similar to the vutrisiran. And this is kind of similar for the cardiac disorders as well. So my question is that can you clarify that whether you think that this is not like treatment related harms or just the characteristics of the population? That's why you said there is not much harm associated with the treatment. And then whether you still think that the harms are pretty similar across the different interventions.
Thank you for this important question. I want to make sure I understand the question correctly. You're concerned in table 10 about the severe TEAE.
Yeah, and then cardiac disorders, yeah.
Yeah, I think the fact that so many of these are seen in the placebo group suggests that they're not effects of the therapy, that in all of these cases, there are more of these bad things in the placebo group, suggesting that this is not an effect of the therapy. I don't know if anyone else has anything else to say in addition to that.
100%. Couldn't agree more.
Yeah, so thanks. Yeah,
I would just also comment that, as has been mentioned, cross comparisons of trials are hugely problematic. So we initiated the ATTR Act trial in 2013. Patients were much, much, much sicker in 2013 with this disease than they are currently, which is why we have certain, I think, observations. That's supported by the fact that New York Heart Class 3 were over 30% in the ATTR Act trial, and they're in the teens in the other trials, and probably why in the same fixed duration, one drug showed a mortality benefit because the event rates were higher as compared to others. So as I think Jason elegantly mentioned, I'd be very cautious about trying to compare hazard ratios across trials because the patients that we see nowadays, thankfully, have much less disease at a time, I personally believe, where therapy is much more effective.
Rachel?
Thank you for the very helpful presentation. I want to make sure that I understand the dosing question, including issues around potential differences in efficacy between varying doses of tafamidis. So For dosing, it looks like patients in the LTE were transitioned to the 61 milligram free acid form, which it says is bioequivalent to the 80 milligram form. But in the trial, there were patients on both the 80 milligram and the 20 milligram form. And so for the clinical experts, I'd like to hear about in clinical practice, what fraction of patients are maybe on the different formulations, the 61 milligram free acid form or the 80 milligram form, which it seems like is delivered in 4 20 milligram capsules. And do you see any clinical differences between the two formulations? And then for either the clinical experts or the ICER team, how should we think about why both formulations are on the market and how, if at all, it should affect our thinking?
I'll take the clinical piece of it, and then I'm going to pass it to Dr. Maurer, who basically created and ran the ATTR ACT trial. So he's going to give us his perspective on the 2080. So in clinical practice, whether you give the 61 or the 80 is just what insurance will cover. Some insurance covers one, some covers the other, and you reassure the patient you don't really care except the inconvenience of 4 pills versus 1 pill. There are some people for whom, of course, cost is a huge problem, and they'll say, well, can I get the 80 and then just ration it and space it out and try to make it work? And I say perfect is the enemy of good. If you can get some, it's better than none. Don't remortgage your house to get it. So from a clinical perspective, it's all equivalent. And in clinical practice, there's no difference I've observed from patients on one versus the other. But I'd be delighted to hear Dr. Maurer's perspective, having created the trial, on what his perspective on 20 versus 80.
Yeah, I completely agree with Michelle. You know, the 61 milligram and 80 are just different formulations. The bioavailability of tafamidis meglumine, which Jeff Kelly invented in his laboratory, and was a standard that we used in the ATTR ACT trial. So 20 milligram pills they're very large, and it was not very bioavailable, which I went into big pills. And when the trial was successful, Pfizer reformulated tafamidis, what we call free salts, and no meglumine, and as a result, we could put 61 milligrams in a single pill that's easier to swallow. You know, and I agree that the approved dose is 80 milligrams or 61, and I agree with Michelle. There are patients who, unfortunately, for cost issues, do need to ration. It's generally believed, I would say, though, that more stabilization is clinically better, hence the reason why acoramidis, which may be at least in vivo, in vitro, I should say, a better or more potent stabilizer, may have more efficacy. But the clinical evidence that emerges from the trials about that is scant at this point in time, in my opinion.
Thank you. Gregory.
Thank you very much. Jason, a beautiful presentation. I wish I could have given it myself. So thank you for that. You referred to these treatments as being disease modifying several times. And yet my understanding is that the therapies don't remove amyloid from the myocardium. And I wondered if, first of all, you could confirm that I'm correct about that presumption. And then secondly, just to extend this, amyloid can be removed from the brain, different amyloid, of course. Is there any anticipation that somewhere in the future it might be possible, for example, monoclonal therapy, to actually remove amyloid from the myocardium?
Thank you, Dr. Griffin, for your kind words. Yeah, just to address the presentation issue, I was trying to get at a term that made the distinction between these agents and like furosemide, you know, medicines that simply decongest. But yes, that's absolutely right. They don't remove it from the heart. I normally pass that to the clinical experts, so I'm confident in saying that. And then maybe I'll defer to them about the questions you had about emerging therapies regarding other organ systems.
Yeah, sometimes I use the term disease-directed therapy just to say exactly as Jason said, it's not a diuretic or guideline-directed medical therapy for heart failure, but amyloid-specific disease-directed therapy. And then I'll let Matt have the exciting role of telling you about the super awesome holy grail of monoclonal antibodies in reversing disease.
Yeah, so completely agree. There are phase 3 trials and one phase 2 trial that have initiated with monoclonal antibodies targeting misfolded TTR. That's so that's and there's a recent publication by a colleague, Pablo Pavia Garcia, in the New England Journal about a phase 1 antibody published last year that demonstrates, at least on surrogates of extracellular volume on MRI, cardiac biomarkers, and scintigraphy, that there is the potential to remove amyloid and reverse the part of the phenotype. So we are very excited about that, but time will tell.
Thank you. Are there any other questions by the Midwest CPAC? It's 10:51 here. I think we should go ahead and start the evidence of the economic modeling. So I'll go ahead and introduce Aaron Nguyen. Aaron?
I was muted. Sorry about that. Hi, I'm Aaron Nguyen. I'm an associate professor at the University of Illinois, Chicago, and I'm happy to talk with you all about the value of these disease-modifying therapy for patients with ATTR-CM. Next slide, please. So I'd like to thank my team members, which includes Dr. Shaw, Dr. Kim, and Dr. Touche here at UIC. Additionally, our collaborators at ICER, including Dr. Richardson and Lee. Next slide. So of this talk today is to discuss the lifetime cost effectiveness of stabilizing agents added to best supportive care compared to best supportive care alone for the treatment of ATTR-CM for adults in the US. I'd like to point out we are not considering silencing agents. The newer agent that was, just had its big publication out last month. Currently, there isn't enough data available to populate an economic model to estimate the value of this therapy. However, we look forward to doing that in the future as data becomes available. Next slide. So to have a sense of how patients are faring, patients with ATTR-CM, we look at the unmet need. And here we're showing the absolute equal value life year shortfall of about 5.5 years. So for patients that have access to stabilizing agents compared to similar patients of a similar age, gender, we estimate about a 5.5 absolute shortfall in equal value life years. These patients are quite old. That's a proportional equal value life year shortfall of almost 2/3. So clearly there's still lots of work to do in this area. You could compare this to other diseases such as multiple sclerosis, which has a much larger absolute shortfall, but a smaller proportional shortfall. And this is just based on the age of the patients. Next slide. So let's talk about the methods for this specific project. So for this study, we used a cohort Markov model. This setting was the perspective that we focused on was the healthcare sector perspective. However, we do scenario analysis that also look at the modified societal perspective. The time horizon is over the lifespan. The discount rate was 3% per year for costs and outcomes. Our cycle length was 6 months. And the primary outcomes that we will show would be cost per quality adjusted life year gain, cost per equal value life year gained, and time spent in class 1 or 2, which Jason detailed earlier what these classes entail. Next slide, please. So we're going to be going through how this model was constructed and how we then use it to estimate value. And these models often have a lot of different details and components that go into them. And so I'd like to just kind of say, give you kind of big kind of chapters, if you will. So first, we're going to figure out the population that we think we should simulate. Then we're going to talk about how we're able to mathematically create and estimate disease progression and what goes into that. And then once we have our population, we've simulated them, we've simulated what their disease progression would look like, then we can attach outcomes to their disease progression. And so we'll talk about these kind of landmarks as we move through these slides. Next slide, please. So first, talking about the population. Jason did a great job walking through these different trials and how the population has changed. And just to reiterate that one more time, the tafamidis trial that took place about a decade ago, patients on average were about 74 when they were diagnosed, and many patients were in class 3. If you look at NYHA functional class, about 32%. The more recent trial for acoramidis, the other stabilizing agent, patients are about 4 years older, 77. We see far fewer patients in class 3. Rather, we see more patients in class 2 and class 1. And so when thinking about the population that we should be simulating, we want it to be close to current practice. And so we're simulating a population based on what was observed in the acoramidis trial. So patients' average age entering will be 77, for example. 10% will enter the model in class 1, and so forth and so on. So that's the population that we'll be simulating. Next slide. And so now we move from that initial component of the population to figuring out how are we going to have patients progress? How is their disease going to progress? How can we describe that? And so based on what has been done previously in the field and what was measured in clinical trials, we're able to model patients' disease progression based on NYHA functional classes. And so what that means is patients, a cohort of patients similar to those found in the acoramidis trial will enter the model, and each 6 months they can move from different NYHA classes or they can die. Them moving between different NYHA classes, we can estimate that based on publicly available data that is based on the tafamidis clinical trial. So tafamidis, that study published transition probabilities both for those using the drug and for those that were receiving best supportive care. And so that is how we're able to capture disease progression for these patients. Next slide, please. So as I just said, treatment efficacy is defined by observed progression through NYHA functional classes. And of note, we only have information on this, on these transition probabilities between NYHA functional classes from the tafamidis trial. We don't have similar information from the acoramidis trial. So therefore, we will be modeling this as a class effect as opposed to individual treatments. We won't have, say, an estimate for tafamidis and then a separate estimate for acoramidis. Rather, it's just at a class level. Next slide, please. So when we looked at that schematic earlier, patients moving between NYHA functional classes, we were able to get a lot of data to populate our Markov model from the tafamidis clinical trial. However, we have less detailed information from trials for survival. Rather, what we do have would be overall survival from patients in the trial. And so we're going to use this as a guide to estimating survival. And one thing I'd like to point out before I go through the details on how we estimated survival is patient survival was very, very similar until 18 months. And then we start to separate. And we're going to incorporate this here in how we estimate mortality. So if we could go to the next slide. So how we estimate mortality is we first start with the US population. the life tables that are provided, and we estimate survival for those that would have been in the acoramidis trial. Average age 77, about 90% male. Then we inflate mortality based on general estimates of the increased risk of death based on what sort of NYHA class. Now, these are based on estimates for patients with heart failure, not ATTR-CM specifically. So we expected that these would be lower, so mortality wouldn't be as high as one would expect for this clinical population. To estimate that, we then take our estimates and we calibrate them. So we directly adjust our survival rates based on that graph we just saw. So we first estimated an inflation factor or hazard rate between 0 and 18 months that was consistent for the treatment group and the comparison group. Then we estimated a different hazard rate or inflation factor for the comparison group, which is 2.75 as listed here. And then we estimated a treatment effect compared to the comparison group of 0.44. And so when we do that, we get very similar estimates of survival as seen in that previous graph. Okay, next slide, please. So far, we've talked about the population. We've talked about how we're doing estimating disease progression. And now I'm going to talk about the outcomes that will be attached to patients as they move through this model. So utilities, costs, and hospitalization inputs were dependent on NYHA functional class, but they were not treatment dependent. So for example, there's a risk of a patient being hospitalized when they're in NYHA class 3. That does not vary whether a patient is using tafamidis or acoramidis compared to just best supportive care. It is purely based on what functional class a patient is in. The price of treatment is based on current tafamidis prices. We don't know what the price of acoramidis will be, so we're just using the tafamidis price. Patients discontinue when they progress to class 4, and we did not model adverse reactions as they were consistent between treatment arms as Jason just discussed. Next slide. Just wanted to highlight our health utilities were based on data provided from the tafamidis trial. However, we had to normalize them to national averages. So what that meant is for NYHA class 1, the tafamidis trial had estimates of a patient's facility that was higher than that of the national average for patients of that age. So we directly adjusted all of these utilities down by the same amount to make sure that NYHA class 1 was not, didn't have a better utility than the national average. Next slide. So as done with economic models, we do a series of one-way sensitivity analyses, which means we see how sensitive the results are to changing one thing at a time, increasing it or decreasing it. We also do a probabilistic sensitivity analysis where we let all parameters vary at the same time based on distributions. And then we also have a series of scenario analyses. And the two that I want to highlight here is one, the societal perspective, which included patient and caregiver productivity costs. Additionally, the other scenario analysis, the costs of reduced dose. So we also do an analysis where we reduce the cost of the drug to 25%. So this is patients going from that 80 milligram or bioequivalent to 80 milligram down to a 20 milligram dose. And so that reduces the cost, as you would expect. We do not change the effect estimates, though. So those should be interpreted, understanding what assumptions are underlying that. If we could go to the next slide. So now the results. So for the base case results, we find that using a stabilizing agent plus best supportive care has a cost about $850,000. QALYs of 2.9, equal value life years 3.2, and years in NYHA class 1 or 2 of 2.7. For best supportive care, the costs are $76,000. QALYs, or quality adjusted life years, are 2, equal value life years 2, and years in NYHA class 1 or 2 would be 1.8. This results in an incremental increase of costs of over $780,000, an incremental gain in quality adjusted life years of 0.9, and an gain of equal value life years of 1.2, and an additional 0.9 years in NYHA class 1 or 2. Next slide. Then when we estimate the incremental ratios, we find the cost per QALY gained using a stabilizing agent and best supportive care compared to best supportive care alone is $873 per quality adjusted life year gained, and the cost per equal value year gained is $627,000. Next slide. When we look at our one-way sensitivity analysis, if you see the x-axis that's at the top, we see that varying our input parameters did not have a substantial effect on the incremental cost effectiveness ratio. It never goes below $850,000 per quality adjusted life year gained. Next slide. When we do a probabilistic sensitivity analysis, we do not find it's cost effective in any case for any threshold, $50,000 per quality adjusted life year gained. Next slide. When we look at our scenario analysis, the modified societal perspective actually results in a increased cost per QALY gained, as we are incorporating patient and caregiver costs. It actually results in a less favorable QALY. We see the cost per equal value life year gained at $731,000. Next slide, please. The other scenario analysis of note would be the cost of the reduced dose while maintaining a similar efficacy, and we find the cost per QALY gained at $250,000 and the cost per equal life year gained at $179,000. Next slide. When we look at the net benefit price benchmark, currently the WAC, or the wholesale acquisition cost for tafamidis, is about $270,000 to reach an annual price of $100,000 per QALY gained. The drug price would have to go down to $13,600 for an annual price at $150,000 per equal value life year gained. It would have to go down to $39,000. So that's a price reduction of 85 to 95% roughly. Next slide. So this study, of course, had some limitations. The primary limitations being we had a lack of available data to inform a differentiated effect between acoramidis and tafamidis. There's limited ATTR-CM specific cost or mortality data, and there's uncertainty on disease progression in ATTR-CM. We're just relying on this tafamidis clinical trial data that has been produced. Next slide. We received comments looking at mortality estimation and hospitalization rates. There's concerns about the accuracy of our mortality calculation. So we changed how we did this. We did this two-phase approach where we first look at the first 18 months, which is a consistent mortality between the treatment and comparator arms, and then we had it separate. So that was one change we did based on comments. Additionally, there were comments about bias in the initial hospitalization rates, which we were having different based on if a patient was in the treatment or comparator arm. meaning if you were an NYHA class 3 and you were using the treatment, you may have a different hospitalization rate than a similar person, still NYHA class 3, that was not using a treatment. So we no longer had differential hospitalization rates by if a patient was using a treatment or wasn't. Rather, we used pooled estimates from the tafamidis clinical trial. Next slide. So what we find is stabilizing agents plus best supportive care generate greater length of life and quality of life with much greater costs compared to best supportive care alone. The cost effectiveness of stabilizing agents plus best supportive care exceeds commonly used cost effectiveness thresholds in the US. Next slide. Now we open it up for questions.
Aaron.
Yeah, thanks for a great presentation. I wonder if you could just explain a bit better how the cost goes up when we consider the societal cost per quality, because it would seem like if there was any extra benefit at all, the cost would go down, but it seems like using this drug costs society money.
So this is the societal perspective is incorporating why we see this difference is because it's incorporating caregiver costs as well. So patients are living longer and caregivers are providing more time to care for them. That's the primary driver on the difference.
So it's actually that it's like we have to spend more because they're living longer to provide care.
Yes.
Okay.
Brad.
Hey, hello, Aaron, and a UIC alum. So glad to hear presentations from UIC faculty. I guess I'll make a comment and then allow you to respond. But it seems like a lot of the modeling decisions kind of may potentially dilute the effect of these drugs. Like pooling the 20 and 80 milligram data together, where there may be hints at greater efficacy at 80, not incorporating using NYHA pooled estimates for hospitalization rates, which the trial data show that there's better hospitalization rates within some of the NYHA classes for these drugs. The utility estimates are driven by NYHA status, but there's quality of life data, and we don't have if you're an NYHA 2, that there's a utility benefit. There's the potential for that. I guess just a lot of these decisions weren't explored through sensitivity analyses. So I just wanted to give you an opportunity to kind of think about or maybe respond to some of the thinking as I think about the model. Now, having said that, is this all these decisions, are they gonna pile on to a cost effectiveness ratio that's gonna get anywhere close to 150,000 per quality? No chance. But just
Sure. So initially we did build the model with different utilities, or we had iterations where the model was built with different utilities based on treatment arms, different hospitalization rates based on treatment arms. And as you said, this has a very small impact on the end of the day result.
Just to go a little further, Aaron, does it improve or worsen when we do it that way? Because my recollection is that because of some oddities in the data, it actually looked worse for the drug when we had it looking like that.
That is true.
David.
Yeah, Aaron, great. Thanks for the great presentations. I have 2 questions. And first, in your Markov model figures that describes how patients progress through these NIHA classes, it has arrows actually indicating patients actually improving. So like moving from like NIHS class like higher to like lower, which means like they're improving. So I don't know whether that was mistakes in the figures or that you actually modeled that it's a possibility of a patient's betting like moving up to like better health state. And if so, then how you actually estimate the treatment effect on the progressions to like better health state? So that's my first questions. Second question is that when you explain about your modeling mortality benefits, so I heard that up to the 18 months, you assume that there's no survivor benefits, but beyond the 18 months, you apply the survivor benefit. I assume it's a 0.7 hazard ratio based on those initial trial. So do you apply the same 0.7 hazard ratio beyond the 18 months? Because as we see in this the other clinical trials for the vutrisiran, if you're actually looking at like long-term horizons, then you may see like greater survival benefits. And also it's shown on the Kaplan-Meier curve that you kind of, they're like the gap actually splitting up. So were you able to, or at least explore the modeling greater long-term survivor benefits as you extend the time horizon, or are you just sticking with 0.7 reductions just beyond the 18 months?
Sure, great questions. So first to talk about what the Markov model looked like. So we were able to use data that provided these transition probabilities that were broken out actually over time. So this allows for variation in how quickly patients' disease is progressing. And yes, the clinical trial did show that patients at times were moving backwards, meaning they could go from, say, an NYHA class 3 to a class 2. We didn't see jumps from, say, class 4 to 1 based on the clinical trial. But you know, they measured everything and they would have allowed for that. So yes, we did see patients' disease progression go in some ways reverse, and we allowed for that in our model. And that is purely based on the trial data. Second, when talking about the mortality effect, we did not use the 0.7 from the clinical trial. That was looking over a longer time frame. Ours instead, we only allowed for separation of the mortality effect at that 18-month time point, which resulted actually in a much larger hazard of about 0.5 or 0.4. I think it's 0.44 if I recollect. We then examined how this fit to additional data that was published where, you know, randomization was somewhat broken in the extension part of the trial, and we could still match survival relatively well. So our estimates were quite close to what was also being estimated. So that is how our mortality effect was estimated and how we essentially validated it.
Matthew.
Yeah, thanks, Aaron. And just want to put some clinical context from my perspective about this. These are always very difficult analyses to do, and we did one with Drew Kazi and got very similar numbers. I would say this is a therapy that, at least in the clinical world, gets better over time because we're diagnosing people earlier. So if I go back to the ATTR ACT trial, while these point estimates are just estimates and the confidence intervals are wide, to give you perspective, the rare patients in the ATTR ACT trial who were New York Heart Class 1, there were only 37 of them out of the 441 had a 64% reduction in mortality. Class 2 had a 39% reduction in mortality, and Class 3 only a 16% reduction in mortality. And we reported that those individuals who were Class 3, to be frank, probably were Class 3B to 4, and investigators were just encouraging to get their patients in a trial. They were pretty sick, which is probably why we saw them living longer but having more hospitalizations. The curves that you point out diverge at 18 months. We don't see that in any analysis of ATTR ACT when we break them out by New York Heart Class, and the emerging data from stabilizers and silencers suggests that the curves diverge earlier because obviously less sick patients, and I think that's been a misinterpretation in some ways in the clinical world of the data that it takes 18 months for the drug to work. It doesn't. There are patients in those curves who don't benefit, or if they are, it takes a long time to see a mortality benefit. The other part of this that's somewhat complicated, I think, is that the disease is pretty indolent for years in class 1 and maybe early class 2. But once someone's on a slippery slope, there's a recurrence of a lot of events. And so probably, I don't know how this is modeled, but those who have advanced disease are likely to have more problems over time, whereas those who have early disease. So the main message I would say is, well, I don't support the price at all. I think this is an unusual therapy in that its efficacy in all these therapies are better in early stage patients. And thankfully, the community has engaged. We have non-invasive ways to make the diagnosis. And I think we're seeing those clinical benefits in patients quite a bit.
Are there any other questions from the Midwest CEPAC? Okay, not hearing any, I guess I'll go ahead and introduce for public comments. We have one person, Mariana Bruno.